This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is the TCA cycle? Answer: the TCA cycle, also known as the Krebs cycle or the citric acid cycle, is a series of chemical reactions that occur in the mitochondria of eukaryotic cells and in the cytosol of prokaryotic cells. It is the primary source of energy for the cell, producing ATP and other high-energy molecules through the oxidation of acetyl-CoA. Question: what are the steps of the TCA cycle? Answer: the TCA cycle involves a series of 8 enzymatic reactions, beginning with the condensation of acetyl-CoA and oxaloacetate to form citrate. This is followed by the sequential removal of carbon atoms in the form of CO2, the production of ATP and NADH, and the regeneration of oxaloacetate to begin the cycle again. Question: what is the purpose of the TCA cycle? Answer: the TCA cycle serves several important functions in the cell. It generates high-energy molecules such as ATP and NADH, which are used to power cellular processes. It also produces CO2, which is used in the process of photosynthesis in plants and other organisms. Additionally, the TCA cycle intermediates are precursors for biosynthesis of other important molecules. Question: what are the intermediates in the TCA cycle? Answer: the intermediates of the TCA cycle include citrate, isocitrate, alpha-ketoglutarate, succinyl-CoA, succinate, fumarate, malate, and oxaloacetate. Question: what enzymes catalyze the reactions of the TCA cycle? Answer: the reactions of the TCA cycle are catalyzed by a series of enzymes, including citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, succinyl-CoA synthase, succinate dehydrogenase, fumarase, malate dehydrogenase, and pyruvate dehydrogenase. Question: what is the role of NADH and FADH2 in the TCA cycle? Answer: NADH and FADH2 are produced during the TCA cycle as a result of the oxidation of intermediate molecules. NADH is produced in the form of NAD+ through the action of isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase, while FADH2 is produced through the action of succinate dehydrogenase. These molecules are then used in the electron transport chain to produce that. Question: how does the TCA cycle relate to cellular respiration? Answer: the TCA cycle is a key component of cellular respiration, which is the process by which cells generate ATP through the oxidation of fuel molecules. The TCA cycle takes place in the mitochondria and is closely linked to the electron transport chain, which generates ATP through the movement of electrons through a series of protein complexes. Question: how does the TCA cycle relate to metabolic pathways? Answer: the TCA cycle is a central metabolic pathway that connects with many other pathways in the cell. For example, the TCA cycle intermediates are precursors for the biosynthesis of amino acids, nucleotides, and other molecules. Additionally, the TCA cycle is closely linked to the process of glycolysis, the breakdown of glucose to generate energy, and the process of gluconeogenesis, the synthesis of glucose from non-carbohydrate precursors. Question: are there any disorders associated with the TCA cycle? Answer: yes, there are several genetic disorders that can affect the TCA cycle. These include defects in the enzymes that catalyze the reactions of the TCA cycle, leading to a buildup of toxic intermediates and a deficiency in energy production. Some of the disorders associated with TCA cycle are Lee's disease, mitochondrial encephalomyopathy, lactic acidosis, and stroke-like episode. Question: how does the TCA cycle regulate its activity? Answer: the TCA cycle is regulated by both allosteric and covalent modifications of its enzymes. For example, the activity of some TCA cycle enzymes is inhibited by high levels of their products, such as NADH and ATP, which act as allosteric inhibitors. Other enzymes are regulated by covalent modifications, such as phosphorylation or dephosphorylation, which can turn the enzyme on or off. Additionally, the TCA cycle is also regulated by hormones such as insulin and glucagon, which can modulate the activity of the enzymes involved in the cycle. Question: how does the TCA cycle integrate with other metabolic pathways? Answer: the TCA cycle is an important hub in the metabolism of the cell, linking with other metabolic pathways such as the citric acid cycle, the electron transport chain, and the urea cycle. The cycle's intermediates are also used as precursors for the biosynthesis photosynthesis of many important molecules such as amino acids and lipids. Additionally, the TCA cycle is also connected to the process of gluconeogenesis and glycolysis, which are responsible for generating glucose and ATP respectively. Question: can the TCA cycle occur in the absence of oxygen? Answer: no. The TCA cycle requires oxygen in order to produce ATP through the electron transport chain. The TCA cycle is an aerobic process and is dependent on the presence of oxygen for the final stages of the cycle to occur. In the absence of oxygen, the cell will turn to anaerobic metabolism, such as fermentation, to produce energy. Question: is the TCA cycle present in all organisms? Answer: the TCA cycle is present in most eukaryotic organisms, as well as some prokaryotic organisms. It occurs in the mitochondria of eukaryotic cells and in the cytosol of prokaryotic cells. However, some organisms, such as some archaea, lack the TCA cycle. Instead, they use alternative pathways to generate energy.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the First Aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.